Okay, started. All right, people. So let us move on. So we have discussed two different rebuilding. Two approaches in systematics: phonetic approach and cladistic approach, right? And at the end, we decided that the cladistic approach is better because it is based on, it uses evolutionary homologies, and it's true for, at least the cladists would argue that what you get out of a cladistic analysis is true phylogeny, its evolutionary relationships, whereas the phonetic approach, it's more of a clustering algorithm. That is just clustering entities based on overall similarity. So the phonetic approach gives you some idea about how species might be related, but it is, strictly speaking, not a true phylogeny. Okay? Now we'll go over some examples to illustrate the importance of synapomorphies. Okay? And what happens when you're not using the right kind of characters. Okay, here's a phylogeny of human and great apes. So my question to you is this the right phylogeny? No. So in this phylogeny, humans are sister to all the apes. Where should humans fall? Along with chimpanzee, right? But for the longest time, people thought this was the right phylogeny. Why? Morphological overall similarity. If you look at overall similarity, these things look very similar to each other, and we humans look very different from them, right? So for the longest time, people thought this was the right phylogeny, right? In fact, humans were placed in a different subfamily, I believe. But then what happened was genetic data started showing something different. The earliest work was with DNA hybridization work. Even before that was looking at blood proteins. All those studies started showing that humans were actually quite closely related to the great apes. Okay, great apes are these three. I'm sorry, these three, right? Not the gibbon. So that was sort of an embarrassing, at least to some humans, it was rather embarrassing because we are so unique, we are so different. How can we be like apes, right? But as more and more genetic data began to accumulate, what basically we realized was that humans are actually sister to chimps. Now we have complete genomes, and the results are actually even more embarrassing because basically what it's showing is that the difference between humans and chimps is just 1%. Right? Most of the genes are pretty much identical between humans and chimps. So what has happened here is there have been many unique characters that have been acquired on the lineage leading to humans. And there has also been loss of many characters, thereby making these entities look more similar to each other. Right? So that's why overall similarity can be problematic. And that's why using synapomorphies or evolutionary homologies is important. Okay? So this is the character matrix that has resulted, that would suggest that humans are very unique and different from the rest. Okay? So can you think of some characters that have been lost on the lineage leading to humans?
Body.
Yeah, body hair. So we don't have as much body hair as
Canines.
Huh?
Sharp canines.
That's a good one. I had not thought about that. But do all apes have sharp canines?
They have larger canines than us.
Oh, okay. Okay. What about unique characters on the lineage leading to humans? Huh? Language, well, one might argue that even chimps and gorillas have language. It's just that we don't understand, they understand each other. But cognitive ability, everything to do with brain. So we have a larger brain. And
advanced vocal cord.
yeah, that also, yes. But because of large brains, a lot of other things follow up with that. Tool making. Actually, chimpanzees also make tools. But large brain is something quite important character that is unique to humans. And obligate bipedalism, right? Chimpanzees sometimes do walk on two legs, but we always are walking on two legs, right? So basically, those are unique characters, and there have been a lot of character loss. So thereby making humans look very different. So overall similarity can be problematic. Here's an example of convergence. This is a New World vulture. New World as in North and South America. And this is an Old World vulture. They look very similar. And they were put in the same family and all that, along with the other birds of prey, other raptors. These are storks down here. And what is amazing is when people looked at the molecular data, turns out that the New World vultures are related to storks. This is one of the earlier studies, right? This is a more recent one, but anyway. So the New World vultures are related to storks, right? And look at so that means this fellow is more closely related to these than to that. Morphologically, they look very, very, very similar, right? So this is convergence. Homoplasy, right? Similarity because of independent acquisition of the same character. Not similarity because of shared ancestry. Similarity because of shared ancestry would be synapomorph. Evolutionary homology. This is homoplasy, right? So overall similarity can be really deceptive because of such cases, right? The convergence here is quite remarkable in the sense that the New World vultures are also carrion feeders, so they feed on dead animals, and they have got all the adaptations that the Old World vultures have to feed on carrion, right? And they also, so have hooked beak. The New World vultures also soar in the sky, so they basically use thermals to glide. The New World vultures do that, the Old World vultures also do that, right? So this is it's remarkable convergence, okay? So overall similarity can be problematic for these fields, okay? So this brings me to these terms that people often use in taxonomy, and that is monophyly, paraphyly, and polyphyly. Okay. So what is monophyly? Well, a monophyletic group consists of all descendants of an ancestral taxa. Okay. Basically, a clade is a monophyletic group. So one way to look at it is if you are comparing traditional taxonomy with molecular phylogeny. Okay. In traditional taxonomy, say a particular genus has 3 species, A1, A2, and A3, right? And another genus has 2 species, B1 and B2. Now, you would expect them to cluster together if you build a molecular phylogeny, right? So this is linear taxonomy, and you build a phylogeny, and it turns out A1, A2, and A3 branch together. So basically, genus A is monophyletic. It consists of the ancestral taxa and all its descendants. right? So this is when you're comparing molecular phylogeny and morphology-based taxonomy. So here there is no conflict. Genus A is monophyletic, genus B is monophyletic, and a monophyletic group is called a clade. Okay, so it has the ancestor and all the descendants. Here is an example where there is a conflict between molecular phylogeny and morphology-based taxonomy. Okay, genus A is not monophyletic here because nested within what we are calling genus A is another species from a different genus. Okay, so this is something called paraphyly. A is paraphyletic. Okay, can you think of an example of paraphyly that we have already discussed? Yeah, apes. Apes are paraphyletic. What we are calling apes is not monophyletic, it is paraphyletic. And in cladistics, we want to define monophyletic groups. Right? Apes are paraphyletic because when we say apes, we don't include humans. Right? And here is a case where these two species are assigned to the same genus, but they are not sister to each other. So what is this? Huh? Louder? Anyone? Example that we already discussed? Convergence, yes. The vulture example, right? So this is polyphyletic. Poly is multiple, and phyly is branch. So it has multiple origin, right?
Does it mean A1 and A2 is polyphyletic group?
No, it simply means genus A is polyphyletic.
Okay.
Right? So genus A has many species. We expect it to be monophyletic like this. Instead, those species are all over the place. So that means the characters that have been used to describe genus A has evolved multiple times in unrelated lineages. Right? So, so we say that, hey, you know, that genus is polyphyletic. We have to revise the taxonomy. There's a problem with this genus.
So let's consider A1 is species 1, and we have multiple sequence data for A1.
Yeah.
So A1 and A2 are belonging to single species.
Okay.
So does it make sense to say that species A and A2, which is one only, that group is polyphyletic?
No, you're saying A1 and A2 are sister to each other?
It's the same species,
Okay.
but we have multiple sequence data for that.
Yeah.
But when we get the phylogenetic tree, it appears the same tree. Let's assume that.
Which tree? This tree?
Third.
Yeah.
So how we can interpret that species which same similar species with many sequence data which appearing like this tree. So that species is polyphyletic.
Oh, you're saying that if A was a species? Yeah. It does not matter. You know, whether you are calling your taxa a species or a genus or a subfamily, it does not matter. Right? It is a term given and the hierarchy it's placed in, all that is based on your traditional taxonomy. Right? So there are many individuals that all share the same character and are placed in a single species, and when you build a phylogeny, some of the individuals go somewhere else, some individuals branch somewhere else. So that species is polyphyletic. And you do see that in some cases. Right? But usually polyphyly occurs at a higher level. At the species level, you don't see a whole lot of polyphyly. I'm trying to think, are there examples of polyphyly at the species level? If I remember, I'll let you know. But yeah.
Does that mean like in the polyphyletic, if there is like mixing of there is exchange of gene pool.
No. It's just a poor taxonomy. That's all.
So if such is the case, basically the taxonomist has to come in the picture and solve.
Yes. So what typically we would do in this case is we would say, okay, one of them, the genus name has to be changed. Okay, and maybe the type for this genus is A1. So this species will retain this genus name, and here you change it, right? And ideally, then what you should do, you should use different characters for these genera, right? Because this character is clearly not good, right? Because it's showing convergence.
But
Yeah.
Nowadays, like the phylogeny is mostly dependent on molecular data.
Yeah.
And the earlier, like the vulture example, was based on the morphological data.
Yeah.
So when we do it with the molecular data, then it is following the monophyletic group
Yeah.
of two different set of vultures.
Yeah.
So how can it be a polyphyletic group? Because we have placed it wrongly.
Yeah. You are absolutely right. These are terms used to describe the discordance between molecular phylogeny and morphology-based classification, right? So in this case, what you are doing, you're saying, okay, B is part of A1, genus A. We have to make genus A monophyletic, so I'm going to assign this species to genus A. Now genus A becomes monophyletic, right? So it is just a way to describe the discordance, and we ideally want this, right? So in this case, we say, oh, genus A1, AA is polyphyletic. One of them has to be ascribed to a different genus, and we go on to do that. So these are just terms used to sort of explain this discordance. Does that answer your question?
Yes. Like if you are going through molecular phylogeny, then they are actually, I feel they are following the monophyletic.
Yeah. I completely agree. Yes. But the point.
I see the contrast in the theory. Like, does the polyphyletic exist in reality?
Yeah.
A bats.
Bats are not polyphyletic. I mean, I don't think so. No, so, I mean, you're right. I mean, in a phylogeny, clades obviously are monophyletic, right? In a phylogeny, you will retrieve something like this.
Because that is the most sparsely.
Yes. But then the entities in that have been identified based on morphological characters by a taxonomist. Yeah. So when you place that name on the phylogeny, then all these things emerge.
Okay. But if you change the genus, then not.
Yeah, of course. Yeah. So that's what here you would change it. You would say A is polyphyletic, so you change one of them. And here we say we are going to subsume B into A. Okay. Yeah.
So it would be easy to picture this in case of cryptic groups, right? So for example, if there is a lot of phylogenetic crypsis, there are actually multiple lineages or multiple putative species.
Yeah.
But at the moment, due to current understanding, they are putting all of them into a single universal species or a genus.
Yeah.
Cryptic species would be a straightforward.
Cryptic species is a slightly different case. Cryptic species is more like what was thought to be a single species actually consists of multiple species. Here, these are different species already.
Taxonomically recognized as distinct species.
Yeah, different species, but have been placed in the same genus. Cryptic species are usually sister species, and when you do the genetic work, you realize, it consists of two clades. There's a deep divergence between those two clades. They're probably two separate species, right? So that's a different situation altogether.
This is an issue which generally occurs most when you're working with fossils or extinct species, polyphyletic. I think this kind of issues have occurred multiple times because those are people have only to work with the morphological characteristics, and a lot of the species or genus where interchange and it has been going on. They have been
Yeah, no, but the point is, this is when you're comparing molecular phylogeny with morphological data, right? So you need, it's just describing discordance, so you need data from both. Whereas in case of fossils, it's only morphology, right? Okay, let's move on. Yeah, so we already discussed this. Okay, there is actually one more school of systematics. Okay, so we talked about phonetic school, cladistic school. Okay, phonetic school, numerical taxonomy, cladistic school, also called as phylogenetic systematics, and there is evolutionary systematics. Okay, this is actually quite a old school from pre-Darwin's times, and some of the modern proponents of evolutionary systematics include people like Ernst Mayr and Simpson, who was a well-known evolutionary biologist. Okay, so what is evolutionary systematics all about? Okay, it's sort of a hybrid between cladistic and phonetic approach. So phonetic approach, overall similarity, it will end up branching all the apes together, and humans are sister. So that's overall similarity. Cladistic approach tries to identify clades as natural units, right? So here human chimp would be one group, human chimp gorilla another group, and so on, right? So here we are interested in monophyletic groups. Now, evolutionary systematicians, so here there is no scope for paraphyly, right? So humans, even though they are very different and we call all of these apes, would now be subsumed into apes, so that apes are monophyletic. So cladists do not believe in maintaining paraphyly. Okay? Whereas evolutionary systematicians, they recognize paraphyly. They use evolutionary trees, but they recognize paraphyly. They are like, paraphyly is okay, it is telling us something important. Okay? So, in case of cladistics, phylogenetic systematics, classification does not fully reflect various aspects of evolution. That is the argument that evolutionary systematicians make. That cladistics does not fully reflect various aspects of evolution. Evolutionary systematicians would say that evolutionary relationships as well as degree of evolutionary change is important. Okay? So, remember, apes are paraphyletic and humans look very different, so they're like, we will use this tree, we will still call apes apes, even though apes are paraphyletic, because by doing that, we are saying that humans have accumulated a lot of changes. Okay? So their whole argument is that the degree of morphological difference between chimp and human is a lot more than between chimp and gorilla. Right? Yeah. So, and apes are more similar to each other than to humans, therefore they are a natural group. Apes are, and they will make arguments like apes are ancestors of humans. Okay? So, cladists would pull their hair if they listen to that argument, that we evolved from apes. Cladists would say we are apes. Whereas evolutionary systematicians would say we evolved from apes. But what is important here is that the evolutionary systematicians identify this whole group as apes, but it is paraphyletic because humans are nested. So they are basically saying this ancestor here was also an ape and then humans evolved from apes. Right? creationists would freak out. They'll say, no, subsume humans into this whole group and call everything apes. But what creationists would say that human evolved from an ancestral character state that was more similar to these things. Instead of saying humans evolved from apes. Does that make sense?
Can you repeat?
Yeah, so the creationists would say that everything is apes, and the ancestral state over here was more like these species. So humans evolved from an ancestor that looked more like what we are calling apes, but the evolutionary systematicians would say humans evolved from apes, right? So that is the difference. And it's just that if you look at the character matrix, it makes sense. I mean, humans are nested within this. Over here, there's all this stuff. So that means this common ancestor was probably this state. So humans evolved from a state that looks more like these, right? But evolutionary systematicians will say, we'll call all of these one particular group. I know it's paraphyletic, I don't care. And I'm going to say this species evolved from that species. So, that's just the two contrasting ways of looking at it. Okay? So that's what evolutionary systematics is all about. In fact, our textbooks, makes this mistake all the time. We evolved from chimpanzees. Chimpanzees are apes. No, chimpanzees are our distant cousins. You don't evolve from your cousin, right? Your ancestor is long gone. They can't be present today. Right? If you evolved from something, they can't be present today. So evolutionary systematics, the argument that they make, if you think about it, from a creatistic perspective, it does not make sense. If you're talking about characters, then it's okay. The character state here is more like this, what we have today. That is a valid argument. But that doesn't mean that we have evolved from chimpanzees, right? Okay. Yeah, so this basically, is the same thing. I'm not going to get into the details here. So as I said, the common ancestor of human and chimp was more chimp-like than human. That's a valid statement. Whereas, what would the so-called apes have retained? No, somewhere I've written what the evolutionary systematicians would say, that humans evolved from apes. Okay, anyway. Issues with cladistics. So remember, phylogenetic systematics is cladistics. Then you have evolutionary systematics, and then you have phonetic approach. Those are the three methods. Yeah, so this is the argument that evolutionary systematicians make against cladistics. That degree of evolutionary change is important. That is, this degree of evolutionary change is important. But the problem here is you're looking at only morphological characters that you can perceive. Obviously, we look very different from chimpanzee when we look at only those characters. What about so many other characters that unite humans and chimpanzees? What about all the genetic data, right? So evolutionary systematicians actually seek to classify organisms based on evolutionary relationships, but also take into consideration degree of evolutionary change. So they are like, by overemphasizing on monophyly, you are missing out on some of these unique changes that are occurring in some of these taxa, which make these other groups paraphyletic, right? But the problem, as I said, with evolutionary systematics is that they make extant species that exist today, can be considered as ancestors, which just does not make sense. It's like saying that your cousin is your ancestor, right? So people have also said things like, similarly, dinosaurs gave rise to birds because birds are nested within dinosaur radiation. It's the same issue as ape and human, right? We should call everything dinosaurs. Birds are extinct flying dinosaurs. Yeah, apes does not include humans, but gave rise to humans. Like apes as a group is paraphyletic. Anyway, so these are some of the issues with evolutionary systematics. In fact, paraphyly is very common. What we call reptiles is not monophyletic. Birds are nested within reptiles. And then you have dinosaurs over here, and birds are actually nested within dinosaurs, right? So paraphyly is actually very, very common. And this brings to fore an important point, and that is morphological data can be highly problematic, right? All of these are called reptiles because they have scales. Birds have feathers, right? When you build a molecular phylogeny, you realize, based on molecular data, you realize birds are nested within reptiles, right? Then we should call all of these reptiles. So now this whole group is called by some term, I forget. So there are synapomorphies that unite that whole group. And it's a monophyletic group, of course. Okay, so this brings to fore an important issue, and that is using morphological data for determining evolutionary relationships can be highly problematic because of homoplasy, convergence, and paraphyly. Right? So that's why we have now moved on to molecular data. Okay, so going back, we have looked at these two approaches. That's phonetic approach, UPGMA tree. It's called a phenogram or a dendrogram. Cladistic approach, it's called a cladogram. If you scale the branches, it's called a phylogram. Here we are looking at tree length, right? And in case of cladogram, remember, branch lengths don't mean anything. It's very important to keep that in mind. In a phylogram, branch lengths do mean something, right? How many changes have taken place. Similarly, in a phenogram is UPGMA. And remember, in a phylogeny, this distance between taxa does not mean anything, right? So this vertical distance, the horizontal distances in a phylogram and a phenogram mean number of changes, but this distance does not. What do I mean by that? If you flip, for example, here there are two cladograms. Are they both the same tree or a different tree? Same tree, right? Over here, the distance between C and D is less. Here it's more. That distance does not make any sense. That's what I was trying to get at, right? What we have, the way to read the tree is to look at what are the species that share a common ancestor, right? A, B, and C share a common ancestor. Whether you flip C over there, actually the phylogeny has not changed. Okay. So quickly, I will. Oh, we have time. Yeah. I just wanted to. Also, any questions here? Yeah. So when you are using these terms, please pay attention to the meanings of those terms. Don't just blindly put down, oh, it's a dendrogram, while you might be showing a cladogram. Yeah. Oh, what happened here? Okay. Okay. Yeah, these are again some terminologies. You can have, slant cladogram or a tangled cladogram. It's basically the same phylogeny. And remember, cladogram branch lengths don't mean anything. It's just telling you how species are related. We can have unrooted tree or rooted tree. A rooted tree has a root, a common ancestor for all the species, and this is unrooted tree. And the most important point is that most tree building methods except UPGM actually give you an unrooted tree. This is a very important point to keep in mind. And then you have to root the tree. UPGMA automatically gives you a rooted tree. All other methods give you an unrooted tree, and then you have to root the tree. Okay? And how do you root the tree? I think I'll just skip this part. Okay, so what is the root? You often say root node. So the root is basically the common ancestor of all the species in your phylogeny. Okay? And there are a couple of other terms that I quickly wanted to discuss, but before that, tell me something. Is this a cladogram? Is it a phylogram? Is this a phenogram? What is it? It's a cladogram? Phenogram.
Sorry.
She says cladogram. Anyone else?
Yeah, phenogram.
Phenogram. Okay.
Because you have a root.
No. All of them are rooted here. This is cladogram, that is phylogram, that is phenogram. What is the difference between a phenogram and a cladogram? Scale is different, but in terms of tree building method, the most fundamental thing. This is phonetic approach, right? Phenogram. Cladogram is cladistic approach. Most important thing is here the branch lengths are different, right? They mean something. There's so many changes. In a cladogram, branch lengths have no meaning. If you scale the branches according to number of changes, then the cladogram becomes a phylogram. Okay? So what we have seen here, it's a cladogram. It's a little confusing because all the species are lined up and they're at the same distance from the root. So you might be tempted to say that, oh, it's a phenogram or a dendrogram, output of UPGMA. But there is no scale. Usually a UPGMA tree, there will be a scale saying that, this is so many changes. So this is a cladogram. Okay. The internal points here are called nodes, and that would be a root node, the root of the tree. And of course, these are all branches. In this case, the branches are not scaled, so that's why it's cladogram. And we often use the term tree topology, which is basically the shape of the tree. When I say that the tree topology here are the same, it basically means, even though the shape is different, the relationships are not different, right? When you say the tree topology is different, the relationships between species have to be different. Okay, let us go to rooting. So as I said, the point on the tree that represents the earliest time in the evolutionary history of all those sequences is the root. It's the common ancestor of all these species. Now, all tree building methods give you an unrooted tree except UPGMA, and you have to root the tree. Okay, and an unrooted tree looks like this. Sorry, it's a little bit off. Okay, and unrooted tree is not as informative as a rooted tree, right? Because in rooted tree, you know the direction of evolution. You know evolution is going from here to here. This is the past, this is the present. Unrooted tree, all it tells you is there's a cluster here, there's a cluster here, and there's a cluster here, and the relationships between those clusters are also not very clear. But once you root the tree, it becomes clear. Okay, question. What is this? Cladogram, phylogram, phenogram. Phenogram. This is a phenogram. All of them are at the same distance, but it does not look like cladogram because these branch lengths are all different. Right? Especially if you compare like two taxa. Right? So it's a phenogram. And there's a scale down here. Okay. So, as I said, most phylogenetic methods produce an unrooted tree because the tree building methods had no means of determining the direction of change. Okay. This will become clear when we do parsimony with unrooted, where we look at unrooted trees. So you have to root the tree. There are two ways to root the tree. So as I said, you know, when you root the tree, the estimate of the phylogeny improves. Remember, unrooted tree, you know, you just know that these are different clusters. I don't know why these things have gone off, but anyway. But how are these clusters related to each other? You know, we don't know. We don't know the direction of evolution. So that's why rooted tree is better. There are two ways to root the tree. There is outgroup method and there is midpoint routing. Okay, outgroup method, you need to use an outgroup which is sister to your in-group. Midpoint routing, I'll talk about it in a minute. Okay, here's an example of outgroup routing. So here's the ape phylogeny, and if you want to root the ape phylogeny, so unrooted ape phylogeny would look like that. Okay. Now, if you want to root this phylogeny, you include. So you know that apes belong to a particular subfamily. And from traditional taxonomy, you know that probably the closest relatives of the apes are these monkeys called Sarcopithecidae. Okay, these are old world monkeys. You know, they consist of baboons and langurs. So we know that these are probably the sister group of apes, and you are interested in determining the relationships within this. So you include these as outgroup. Okay, and the tree will look like this. And when you say that langur and baboon are the outgroups, what the program will do is it will cluster all these together, and the outgroup will be sister to the in-group. Yeah. So these are in-group. These are the species that we are interested in. So you will get a tree like that. Okay, does that make sense? Yeah. Now, if you don't have an outgroup, so in this case, we knew that these two species could serve as an outgroup. But if traditional taxonomy, does not provide you with the information on which is a suitable outgroup, you can do something called midpoint routing. Okay, and what is midpoint routing on the tree? So this is a unrooted phylogram. So that means the branches actually pertain to number of changes, right? So you take the longest distance between two species. It's probably this. Okay, and you place the root at the center of that long branch. Okay, so what do I mean by that? You know, if this distance is say 12 changes, and say this is 10 changes, right? So 12 plus 10 would be 22. You divide 22 by 2, by half, you get the midpoint of the longest branch length between two species. You grab that point and pull to one side, all of that will collapse to give you a rooted tree.
Midpoint routing method can be used in which software?
Every software.
Maximum likelihood and all this, they already ask for now.
They ask for an outgroup. You can change the setting and say that go for midpoint routing. Okay. So, however, midpoint routing, if your data set is evolving in a clock-like fashion, then midpoint routing is okay. Otherwise, midpoint routing can be highly problematic. Okay. So, what do I mean by that? Let's see if this is going to work. Ah, yes. Okay. So. Let us say that this is the true phylogeny. Okay, rooted. Now the unrooted tree, in this case, would look something like this. Right? Which is the longest branch here? Between, so it is from D to A. Okay? And the midpoint of that is somewhere here. Now, if you root it at the midpoint, what is the topology you are going to get? Right? There's your midpoint. So you have pulled the tree from here, all of them. Right? So actual topology is that. So you can get a completely erroneous tree. The same with outgroup also. You have to be very, very careful.
So in that case, what would the undersubgroup?
If so, this problem arises when there's unequal rates along different lineages, very, very unequal. If the rates of molecular evolution is by and large similar, ideally it should be evolving in a clock-like fashion, but that almost never happens. But as long as they're evolving quite similar, rates are quite similar, you can use midpoint routing. Otherwise, it's best to go for outgroup routing.
And what about, for example, in some software like usually there is some already option to go for a midpoint routing. So when I choose that, I know means according to literature there are two A, B, and C, and I know A is an ancestor, but when I opt for a midpoint routing, it shows like B is the ancestor to C and A.
So
How to do in that case? I feel like,
yeah, there's huge difference in rates. That's why it's messing up the tree, right? The best thing to do is hopefully you've got that data set with you. We can actually look at it, what exactly the program is doing. The main take-home message here is routing is extremely important. So you have to do it right. If you don't do it right, you'll completely mess up the tree. If you use the wrong outgroup, that'll mess up the tree. If there's rate heterogeneity and some lineages are evolving rapidly and you use midpoint routing, that's also going to mess up the tree. So pay attention to this. And also you have to find out what the program is doing. Often the program decides what's the outgroup and spits out a tree. You need to go and find out what exactly the program has done. Many programs use the first sequence in your alignment or the last sequence in your alignment as an outgroup, right? And maybe it's not the outgroup. So you'll get a completely messed up tree. So pay special attention to routing. Okay, so we'll discuss this more. Yeah, you had a question?
So, outgroup is theoretically its closest relative and belonging to almost same geographical region.
Need not be. Need not be. So outgroup ideally is sister to your in-group. Okay?
Can be of same family?
Yeah, it depends on what what taxonomic hierarchy you're working at. So if you're
If it
looking,
is for the species,
huh?
then the outgroup will be, if it is for the genus, then
Huh.
the outgroup will be of other genus
Yes.
of the same family.
Yes, yeah. Ideally, that's the way to do it. Ideally, a sister genus, of course, in the same subfamily or family, but we don't know if that genus is a sister unless you have a larger phylogeny. So you basically pick another genus in the same subfamily or family, right? And you're also assuming that your genus is monophyletic. So there are all those issues, right? You have to keep trying it out and figure out what's the best approach. There was something I want to mention here.
Also, does the number of outgroups make a difference to your tree?
Number of what?
Outgroups that we select, like 2 or 3 outgroups or 4. How do we decide that?
So sometimes it provides more stability to the tree if you include more outgroup. Yeah.
But not much difference.
It really depends. This is again something that you have to try multiple times to try and figure out. Try different outgroups, and so you can have outgroups that are monophyletic. So outgroup will be. So that's the outgroup. So outgroup can be monophyletic, or in this case, there are 2 outgroups, but the outgroup is not monophyletic. So sometimes these things help in bringing more stability to the phylogeny. So it's a good idea to play with different outgroups. Now, the most important thing is outgroups should not be very, very distantly related. Because then what happens is you'll have so here's your in-group. And the outgroup is like that. Very, very distantly related. So the relationships here will kind of get muddied because most of the changes in the phylogeny is over here, between the in-group and the out-group. And if the outgroup is very, very closely related, you have to be a little careful there because maybe it is part of the in-group and you have forced it to be outgroup. Then you'll get a completely messed up tree again, right? So basically you have to play around with outgroup. So we'll discuss this more when we actually start the lesson. It'll make more sense, but pay attention to what the program is doing. Yeah. So, any questions? We'll break in 5 minutes for lunch. It's better to use outgroup method. I would use midpoint only if you are totally unsure what the outgroup is.
So can I take leave? I'm waiting to.
Yeah, go on.
So I'm not clear with the paraphyly. Actually, I have confusion.
What is the confusion?
So we'll see. So in the paraphyly.
Yeah, so let me just switch back to. No. Okay. Yeah.
Please explain the paraphyly. The second one.
This one.
Yeah.
So basically, whatever we are calling genus A is not monophyletic because you have another species which we have not assigned to genus A branching with one of the species in genus A. So it's the same as apes and humans. If apes were to be a group, right, we expect them to be monophyletic. Right? But what is happening is all of this we are calling apes, but we are not including humans in apes. Right? So apes are paraphyletic. They are not monophyletic. If they were monophyletic, this is how it would be. Right? So this is paraphyly. Apes are paraphyletic. So how do we resolve this problem? We say that humans are now subsumed into apes. Now we call all of these apes. So that solves the problem. Right? Okay. Any questions?
Not a question exactly.
I'm sorry?
Not a question exactly. But polyphyletic groups, I think, are what we frequently see in there is mimicry.
When it is what?
Mimicry. Like in butterflies, there's a lot of mimicry. So could that be a reason
That,
for-
yes, I think so. No, but mimicry,
Convergence.
usually most examples of mimicry are, completely, in a way, if it is assigned to the same group, then, one can say that, it is polyphyly. But in many of the mimicry cases are like very, very distantly related, say, butterfly species looking similar, one species mimicking the other species. Now, if those two butterfly species have been assigned to the same taxonomic group, then it would be polyphyly, right? But I think often people are able to figure out that, hey, that butterfly is actually a different family altogether. It's just mimicking this particular butterfly from this other family. So usually, taxonomically, they have not been put in the same group. At least there might be some cases there of polyphyly. But it's the same idea, right? It's the idea that you can have convergence.